Well, hello everyone. Welcome to this podcast for Biocore 383 on the citric acid cycle and reducing power. This is part 2, reducing power and electron transport. Before we continue our discussion, it's probably worth reviewing what we've discussed so far with regard to the citric acid cycle. Remember that pyruvate enters the mitochondrion and then it transfers 2 of its carbons via acetyl coenzyme A into the cycle. As it does this, it undergoes oxidative decarboxylation, creating a molecule of NADH and releasing a molecule of carbon dioxide, CO2. The 2 carbons transferred to acetyl-CoA enter the cycle by being added to a 4-carbon compound, oxaloacetate. This creates citric acid, or citrate, hence the name of the cycle. Citrate enters the cycle, and 2 more oxidative decarboxylations result in the production of 2 more NADH molecules and the release of 2 CO2s. In addition, as the cycle proceeds, a molecule of ATP is generated via a GTP intermediate. Ultimately, a 4-carbon compound, succinic acid or succinate, is formed, and 2 additional oxidations occur, creating 2 more reduced compounds, a molecule of FADH2 and a molecule of NADH. If you just look at the cycle and you look at all of the purple ovals in this figure, what you realize is that the main goal of the TCA cycle, the citric acid cycle, is to produce reduced compounds. This leads to some key questions. First, what will we do with all that reducing power? And secondly, where and how are these electrons that we produce via these reductions coupled to efficient ATP generation? Well, to answer the first of these questions, we need to consider a concept that you learned in chemistry. And that has to do with oxidation and reduction reactions. Now, you remember that we can calculate the standard reduction potential, the standard electrode potential for a redox reaction. And typically, we do this by writing half reactions. So let's consider a reaction that turns out to be important for the discussion of electron transport, and that has to do with iron. Let's consider ferric ions, Fe3+, being reduced to ferrous ions, Fe2+. You can see that we add an electron to the ferric ion to do this, so this is a reduction. In fact, half reactions are always written by convention as reductions. Whether they go in that direction or not, it depends on the standard electrode potential and whether it's coupled to another reaction. Other kinds of reduction reactions we've already talked about, and one of those is shown below, and that's acetaldehyde being reduced to ethanol. And you can see that the aldehyde group, that double bond there, gets reduced to a single bond, and this involves the transfer of 2 electrons, and 2 protons go along for the ride for charge balance. Now, I told you that by convention, half reactions are always written as reductions. To determine which way a redox reaction is likely to go, we often look up the value of the standard electrode potential from a table. So let's consider the reaction that was at the bottom of the last slide, acetaldehyde to ethanol, and you can see that it has a standard electrode potential, E'0 of minus 0.197 volts. Another very important reaction for electron transport is the reduction of NAD plus to NADH. That also involves the transfer of 2 electrons, and the voltage is minus 0.32. So if these 2 reactions are coupled, we can sum those reactions and write the reaction as you see down below. Acetaldehyde plus NADH yields ethanol plus NAD+. To figure out which way we should write the reaction, what should be the reactants and what should be the products, it will always be true that the more positive E'0 will tend to go to the right. And so that's why we write acetaldehyde and NADH on the left and ethanol and NAD+ on the right. All right, so you can look at 2 redox reactions and couple them in this way for any two redox reactions in a table that you're given. And in fact, World of the Cell gives you such a table. It's table 10.2. So here's table 10.2, and this shows standard reduction potentials for redox pairs of biological relevance. And there are several key reactions here that are worth pointing out. First, here is a very important one, NAD plus to NADH that we just mentioned. That has a standard electrode potential of minus 0.32. The other reduced compound that was produced during the citric acid cycle, FADH2, is has a less negative standard electrode potential, as you see here. So NADH likes to give up electrons more readily than FADH2. That's what this tells us. And then let's compare to a couple of other reactions. The first is the one that sets the standard for a standard electrode potential of 0, and that is going from two hydrogen ions being reduced to molecular hydrogen. The end of the line for electron transport, it turns out, is at the very bottom of this slide. And that is taking molecular oxygen and converting it to water. So the molecular oxygen converted to water turns out to be an extremely positive standard electrode potential. What this says is that oxygen loves to be an electron acceptor. And in fact, it's the final electron acceptor during electron transport. The fact that this value is so positive allows essentially all of the electrons that are possible to be extracted from this process during electron transport to be so extracted. And this turns out to generate a lot of protons. As we'll see, the protons are what drive ATP formation during oxidative phosphorylation. Right, so remember, these are standard electrode potentials. These are kind of under standard conditions. Under real conditions, the values might be quite different. It's not always easy to measure those real conditions, but just for the standard reaction of H plus to molecular hydrogen, the E prime for that, the real world value, is actually minus 0.42 volts. So just recognize that the standard electrode potentials are a good guide, but we need to know the actual conditions to get actual numbers to do actual calculations. All right, so we've talked about standard electrode potentials, and now let's make the relationship between standard electrode potentials and free energy, because this is what we really want to talk about, is energy production after all. So we can write the net electrode potential for some redox reaction, as we've seen, by calculating the difference between the standard electrode potentials of the acceptor half reaction and the donor half reaction. The acceptor is the one that becomes reduced, and the donor is the one that gives up electrons, so becomes oxidized. We can relate these to free energy in the following way. Delta G0 prime equals minus N times F times delta E0 prime, where F is the Faraday constant and N is the charge, the total number of electrons being transferred. So let's look at this in the case of NADH. Delta G0 prime for oxidation of NADH can be calculated in the following way. Let's calculate delta E0 prime first. Well, in our case, we can look that up from a table, and it turns out that we can calculate this as positive 1.136 volts. Great, well, we can plug in the numbers. And what we get when we do that is a whopping minus 52.4 kilocalories per mole. And you can see that the N value here is 2 because we transfer 2 electrons in this redox reaction. So that is a big number. And it's worth comparing that to our standard energy currency that we've discussed quite frequently already, ATP. The hydrolysis of ATP has a delta G0 prime of minus 7.3 kilocalories per mole. So you can see that the energy stored as reducing power in the case of NADH is powerful. There's a tremendous amount of energy available through reduced compounds. In fact, this is the strategy that we're going to use to ultimately generate ATP during oxidative phosphorylation. Okay, so we've discussed standard electrode potentials and how they fit into electron transfers. And now let's consider where the various kinds of reactions occur that we're going to talk about that utilize these redox reactions. This is table 10.1 from your text and reminds you where some of these reactions occur. Remember that mitochondria have an inner and an outer membrane, and there's a space between those, and then the interior of the mitochondrion is the matrix. Well, some key reactions that occur in the matrix are, that's where pyruvate gets oxidized, and that's where the citric acid cycle reactions occur. So those are in the interior, the matrix of the mitochondrion. At the inner membrane are a lot of the key reactions that we still have to discuss. Those include electron transport, and we'll see that there are some protons that translocate through the membrane, and these are involved in ATP synthesis. And ultimately then this results in oxidative phosphorylation, the conversion of ADP to ATP. Now, it's not just where they occur, but it turns out there's a very precise spatial arrangement of electron carriers that allows for electron transport and all these other events to proceed, and we'll discuss that in good time. All right, so let's now consider the places and the complexes where electron transfers occur during electron transport. And this is a complicated slide. This is a slide, a figure that shows the standard electrode potentials associated with a number of the electron transporters that are involved in electron transport and oxidative phosphorylation. So our friend NADH is up at the very top. It has the most negative electrode potential. And you can see then that it is part of one of the complexes in electron transport, complex number 1. NADH, given its standard electrode potential, is going to want to give up electrons, and it does so to a series of electron carriers in complex 1. And then those electrons are transported to a shuttle compound, coenzyme Q, and then they're transferred to another complex, which is complex 3. Complex 3 contains coenzyme Q and something called cytochrome C, and electrons are transferred through complex 2, and then they're handed off through cytochrome C to a final complex, complex 4, at the very bottom of this diagram. Electrons are transferred through complex 4, and ultimately they're transferred onto oxygen, which is the final electron acceptor in this process. Now, oxygen, because of its positive standard electrode potential, very positive, makes it a great final electron acceptor. You can also see that there's another complex, and unfortunately they're not numbered in order, but that other complex is called complex 2. It's up at the top there, and you can see that I've written succinate in, because it turns out that succinic acid from the citric acid cycle feeds in here. So one of the complexes associated with the citric acid cycle is actually part of the electron transport chain and is localized at the inner membrane of the mitochondrion. And you can see that that complex, which involves FADH giving up electrons and feeding them into the electron transport system, feeds in a slightly different place in terms of standard electrode potentials relative to NADH. Right, so it's this flow of electrons from NADH and FADH2 down to molecular oxygen that is going to generate the energy we need to create ATP. So let's consider some of the components in these electron transport complexes. Well, there are a number of them. One class of molecules in the electron transport chain are flavoproteins. And these use either FAD or something called flavin mononucleotide, FMN, as a prosthetic group. So one example, an important example, is in complex 1. This uses a flavoprotein with FMN called NADH dehydrogenase, and you can see what that does, right? This transfers electrons from NADH into the system and results then in the oxidation to NAD+. A second class of electron transport complexes are iron sulfur proteins, and these undergo redox of their iron group, and many of these are involved in electron transport as well. Third are cytochromes. These have heme groups. They also contain iron, or they have bimetallic iron-copper centers. Cytochrome C is a great example of this, a very important cytochrome. It's a peripheral membrane protein and is involved in shuttling electrons between complexes, as we'll see. And then finally are quinones. These are non-protein components that move within the bilayer of the inner membrane. Coenzyme Q, a very important electron shuttle, is one of these quinones. And one of the great things about being at the University of Wisconsin is that some of the key discoveries in oxidative phosphorylation were made right here at UW-Madison. And one of those is iron sulfur proteins. So they were discovered by Helmut Beinert. And we have a rich local history with regard to all of these processes. Okay, so actually, let's look at one of these electron transfer molecules that we've already mentioned, and that's a cytochrome. So let's look at it. Cytochromes have heme groups, and what we mean by a prosthetic group is you've got this molecule, and then there is a region of this molecule that is attached to the rest of the molecule by covalent linkages that can coordinate with an iron atom, and that iron atom can have different redox states. So you see the Fe, the iron atom, in the middle of this, hold the little dotted lines show that the iron atom is held there in this particular portion of the molecule. And the other molecules are fairly similar. So here, for example, are quinones. So coenzyme Q is one of these quinones, and you can see that it can actually be in two different redox states via electron transfers. Starting at the top, coenzyme Q, which is also called ubiquinone, can pick up an electron to become partially reduced. It's sometimes called a semiquinone when that happens, and then it can pick up a second electron and become fully reduced to a dihydroquinone at the bottom. And you can see where these reductions occur, and you can see that it converts these double bonds between carbons and oxygens to single bonds. And the reverse reactions can also take place. So you get the idea that understanding how these oxidations and reductions occur helps us make sense of what's going on during this complicated kind of relay race of electron transfer. All right, so by these kinds of changes in redox state, then, electrons can be transferred progressively by a series of reactions from electron carriers at the upper left through the system down to final electron acceptor, molecular oxygen, at the bottom right. Okay, so it turns out that it's probably worth thinking about where these proteins come from. And it turns out that the nuclear genome, the DNA within the nucleus of eukaryotic cells, encodes some of the proteins that end up in the electron transport system in the mitochondrion. But you'll remember that mitochondria have their own genomes, and we said that that was one of the pieces of evidence for the endosymbiosis hypothesis. So this is a figure from World of the Cell which shows the circular piece of DNA which resides in mitochondria, and it encodes some of the proteins that are required for electron transport. And not surprisingly, if you look at these proteins and you look at their amino acid sequence and you compare that amino acid sequence to some of the similar proteins in bacteria, these mitochondrial proteins are quite similar to those in bacteria. So mitochondria encode some of their own electron transport system proteins, and then the nuclear genome encodes the rest. All right, so let's look at where these proteins are arranged in the mitochondrial inner membrane, because this arrangement is really impressive, and in fact it's what allows for this complicated series of electron transfers. So this is a zoomed in view of the mitochondrial inner membrane, and you remember then that it's folded, and you can see that structure here. So what this shows, and we'll talk about some of these components in a little bit more detail a little bit later, is that these complexes, and they're numbered Roman numerals 1, 2, 3, and 4, these are the main electron transport complexes that we've been talking about in graphical form already. You see that they're embedded in the membrane. That's the big brown blobs. And then there are some light blue components. Those are things like coenzyme Q that allow transfer between complexes 1 and 3 and complex 2 and 3. And then there are some round brown blobs labeled C. That's cytochrome C. That's another mobile electron carrier, a peripheral membrane protein that transfers electrons between complexes 3 and 4. In addition, you'll notice that where NADH and FADH2 feed into complexes 1 and 2, respectively. Notice that the final electron acceptor, molecular oxygen, gets converted to water at complex 4. And finally, the other thing that is really important to note here is that there are hydrogen ions with little light blue boxes around them. And you can see that as a result of the electron transfers through these different electron transport systems, that there are a bunch of protons, hydrogen ions, that end up in the intramembranous space, the space between the inner and outer mitochondrial membranes, see there in the center. So the net effect of electron transport is the release of protons into the intermembranous space. This is going to be used in a very specific way to drive formation of ATP through those complicated structures in purple near the bottom of this membrane fold of the mitochondrion. And those are ATP synthases. And we'll talk about how that works in detail in a separate discussion. Now, I just want to remind you that one of these complexes here is actually a citric acid cycle complex, because it's where we get FADH2 from, and that's the succinate dehydrogenase. Succinate dehydrogenase is part of complex 2. Right, so that concludes our discussion. There's one more thing I'd like you to think about, just for thought as you walk away from this. We've been talking about all of these reactions in a mitochondrion, in a eukaryote. But aerobic bacteria can also carry out these very same reactions. And it's worth thinking about where that might be true. So think about, based on the endosymbiotic hypothesis in particular, where you might predict aerobic respiration would occur in bacteria. I'm not going to answer that question for you, but I want you to think about it. All right, well, that's all for our discussion of the citric acid cycle and reducing power, and in particular electron transport. Hope it's been useful. We'll talk again soon.